Hi, everyone. So the paper I'm going to be talking about today is called Host Lipid Transport Protein, ORP1, is necessary for Coxiella burnettii growth and vacuole expansion in macrophages. So I gave a brief overview of this paper last week, but we're going to do a journal club today. So here we go. There we go. So just as a quick overview, the general question that they want to answer is the host protein ORP1 utilized by Coxiella to aid the carbohydrate efflux, so the removal of carbohydrate from the Coxiella containing vacuole, and does this benefit its growth? And they test this hypothesis in both HeLa cells and mouse alveolar macrophages. Underneath this, I have each of the cited authors, Dr. Stacy Gilk being the kind of lead lab investigator, and the two PhD students that likely did most of the manual labor for it. Moving on to the background of the protein of interest. So ORP1 generally regulates the concentration of sterols within membranes of a host cell. So this includes within lysosomes and even across the cell membrane. And it has interactions in the ER as well. It exists in cells in two isoforms, this long isoform and a short kind of truncated S form, and abbreviated with ORP1L or ORP1S respectively. So ORP1L is the kind of main focus of the study, and when I say ORP1 later on, I will be referring to just ORP1L because that is what they studied. And this one is different in that it localized, well, I won't say it's different, but it localizes to the membrane of binding sites of lysosomes and to the membrane contact sites between the ER and endosomes. So it's basically kind of coming into contact between where the CCV is contacting with the ER and is at that location regulating cholesterol within the CCV. And its truncated ORP1S form also can bind to cholesterols, but does not localize to the CCV. So I believe it's just to the nucleus and the cytoplasm. So moving to our first figure, I actually kind of flipped this paper. So I'm going figure 4 and 5 first, and then circle back to 1 through 3. But so figure 4 states that ORP1 is required for full CCV expansion and Coxiella burnettii growth in MHS cells being the murine alveolar macrophages. So looking at part A, that's basically just the Western blot of their knockout and a wild type of the MHS cells. So I believe they used CRISPR to just knock it out. And it seems they did a pretty good job. I'd say that it's a good looking Western blot. Moving to C and D, actually, you know what? We'll move to B. So B shows pretty nicely the difference in Coxiella growth between the wild type with ORP1 and an ORP1 knockout. So you can see, I don't know if my mouse is visible. It seems like it is. So this big red blob is Coxiella, and it's big up with the wild type, and it's very small in the knockout. So that's a very nice kind of qualitative depiction of how it's affecting the growth. And here on the left is the quantitative aspect of that, being that the CCV size in ORP1 null cells is much smaller than that of the wild type, and they just measured it in pixels, which I found to be kind of an interesting measure. But yeah, and then they also directly monitored Coxiella growth, and you could see it's only a fraction of the size of the wild type in the ORP1 negative cells. All right. Yeah. So moving on to the next figure. This figure basically just discusses the the buildup of cholesterol in the MHS cells. So they use a compound called philipin to bind. It's a fluorescent dye that can bind to the cholesterols, and it fluoresces. So that's real convenient for this. So in short, they fill a cell with the philipin and they see where it glows and how intensely. And in the wild type, it's not very bright, and in the knockout, it is very bright, indicating that there is a buildup in cholesterol within the membranes, and it might be affecting Coxiella growth, which in our figure above, it seems like it does. So yeah, so they, again, just to go through the methods, they knocked out ORP1 from the MHS cells and infected them with Coxiella, and then monitored the cholesterol within the membranes over time. All right, and so figures 1 to 3 are basically those same 2 experiments, but done in HeLa cells. And so the figure, yeah, so I kind of combined all 3 just because the results are somewhat very similar, but also kind of different in some very key areas. So as shown in figure 1b, they did the Western blot. It seems to have worked pretty well. So no problems there. But moving and okay, yeah, so that's done. Excuse my kind of jumping around. That's very odd recording this. Yeah, so moving on, I'll move to 3A, and that is the same experiment that I discussed in the past slide, but it is also done in HeLa cells. And you can see there's a drastic change in the philipin intensity between the wild type and the knockout. Knockout being this very kind of dramatic bright orange. So that's a really nice indicator right there. Very nice qualitative difference. But oddly, looking at figures 2B and C, the effect of knocking out the ORP1 gene did not seem to have a very drastic effect, if anything, on the growth of Coxiella within HeLa cells. So that's interesting, and they were kind of scratching their heads on it. And I think actually Tori mentioned had a quick question on the perusal that they can't answer, and neither can I, but that will certainly be an area of further interest. So basically, just to tie it all together, ORP1 alters sterol content in HeLa cells, but is not required for Coxiella burnettii growth. All right, going to conclusions. This is making out to be pretty quick, so I don't really know. Anyways, so going back again, their hypothesis was that the host transport protein ORP1 aids Coxiella in exporting cholesterol out of the Coxiella-containing vacuole, promoting its growth and infection. And they found this to be true in how it aids cholesterol efflux in both HeLa cells and MHS cells, but oddly enough, it only affects the Coxiella growth in the MHS murine alveolar macrophage cells. Right. And then, of course, we have lingering questions that weren't quite answered by the paper. And the first of all that I just alluded to is why in HeLa cells does an increase in sterol intensity not cause issues for Coxiella? They didn't really offer up a reason why. And I guess that might just have to come down to how HeLa cells have just been drastically kind of undifferentiated over time, and they're kind of very homogeneous and like not the best model anymore. So that is likely an explanation. And then also, what might the other isotype ORP1S play during the infection? Yeah. So of course, the next steps would be to compare the 2 isoforms, ORP1L and S, and how does that affect the infectious process and causing Q fever. And the last sentence of the paper, which Dr. Reed had in her template, and I thought I might add as well, and I think it wraps this up pretty nicely. It says future investigations will further elucidate the relationship between Coxiella burnetii infection, ORP1, and intracellular cholesterol transport by comparing the specific efforts of isoforms ORP1L and ORP1S on Coxiella burnetii. All right, that's all I have. This is pretty short, but yeah, if you have any questions, let me know. Hopefully I can answer some of them. Yeah, all right. See everyone in a week. Bye.